Hi, Mireia. Am I saying your name right? I would say Mireia, but
Yeah,
probably that's wrong.
yeah, that's a correct pronunciation.
Oh, really? Oh, I'm glad. I am currently posting your website in the chat and
Okay, great.
the current paper link and also your 2019 paper link. Um,
Okay. Although I'm going to talk only about the latest one.
yeah,
So
but if people want to check out more of your work, they have the opportunity.
okay.
I think it was a really great paper, so.
Okay, great. Thank you.
And now I'm adding the presentation, so it should be working.
Okay.
And now I'm sharing. I'm just telling you what I'm doing because I don't want to feel like I'm not nice. I'm just there's a lot of things you can only do when you start the room.
Okay. No problem.
Share on Clubhouse what this room is about. There's an extra button for that, and then on
Okay.
Twitter. So thank you for coming. Thank you so much. I really appreciate it.
Thank you for the invitation.
And yeah, I know it's kind of a little bit of a hustle to like make the Clubhouse account, but I hope you'll enjoy it and then maybe you'll come back.
Yeah, sure. Let's see.
So, how was your day so far? Everything good?
Oh, good. Quite busy the last days, but all good.
Oh, good. As long as it's not bad busy, it's good busy.
It's good busy. It's quite busy, but yeah, in a good way. No
That's good.
complain.
That's wonderful. Yeah. So we have been looking forward to your talk. I think your research is really interesting.
Cool. Thank you.
We had a few like microbiome talks.
Okay.
One was about also the forest, like after a huge fire and before, and
Nice. So how you get the restoration to a normal microbiome?
so we don't know enough to restore it yet about
Okay.
the microbiome of the soil,
Yeah.
but
It
it
was
was
in
the
the
first study
succession.
where they actually just studied before and after was by
Okay.
chance that they had samples.
Yeah.
And yeah, and then we had a couple related to health, but I think this one, your work is way more systematic. Um, um, so because you did already before a large scale kind
Yeah,
of
and with time we are getting larger sample sizes, so indeed we can get more stable results and be able to replicate better our results.
yeah,
This is what we need.
I think it's really unique that you have such large scale data sets. So
Yeah.
that's really exciting. So yeah, it's really great, and we'll start in around 4 minutes. I'm almost about to ask you all the to do the
Okay.
short interview before we start.
Okay.
It's hard to not like to stop.
Okay.
But yeah, and
Good.
do you? So you're a postdoc right now.
Yeah.
Are you planning to stay in the lab you are for a while, or?
For a while, yes. Then, I'm also looking for positions. But there is still a lot we can do in my current lab, so also looking forward to continue. Follow up on the results we just got.
Yeah, for sure. It sounds like it's a really great and well, I don't know, but based on the work, it sounds like it's a place where you can make a lot of. I don't know. Do people give you recommendations to do a long postdoc or short one? It depends so much on
Yeah,
with
yeah,
whom you
indeed. In
speak.
academia, you never really know what's the best. I think at the end, I mean, if the right positions open at the right place, right time, that's
Yeah.
what would work best. And
One
then also,
professor used
what's
to
I mean,
tell me, "Oh, enjoy your postdoc. It's the best time. You don't, you're not responsible for all the bureaucracy and everything."
yeah, I can understand that,
But on the
but
other
well,
hand, yeah, you need to
yeah,
be.
having some more stability would also be good.
Yeah, exactly. I agree. Like it's so hard to know what's
Yeah,
right. You only know afterward.
yeah,
History is always very easy to deduct what was right and wrong afterwards.
yeah.
Before you never
Indeed, after it's much easier.
know. Yeah. And then it all seems to make sense, right? But it could have been completely different.
Yeah.
So I think we can slowly start and
Okay.
with introductions and so on.
Hmm.
And I think people will continue coming in. But yeah, let's just start and go from there. So, welcome
Okay.
everyone to Science Society, and of course, a special welcome to you, Mireia. It's really an honor having you here, that you take out time of your busy schedule to come here. And before we start, let me introduce you to the audience. Dr. Mireia Vales-Colomer, she is a postdoctoral researcher in Nicola Zegata's lab and at the University of Trento in Italy. And her she did like 2 degrees, microbiology and computational biology, at the UAB in Spain and VUB in Belgium. And then during her PhD at Reyes Lab in Leuven, Belgium, she started doing research in the microbiota gut-brain axis, and she was really very successful doing that and published in 2019 really, I think, very well discussed and very well known paper that was included in Nature Milestones in 2019, where she also was awarded a recommendation for the F1000 Prime Prize, and she she then continued to be really interested in studying the microbiome, and especially how we acquire different microbiomes that are involved in health and disease. So I think it's a really wonderful path and field to be in. So thank you so much for coming.
Thank you. Thank you very much for the introduction.
And as I said before we start, we do a short interview. So how did you how did you realize that you wanted to become a scientist that you are now? Was that something you always wanted to do, or does did the interest for science become came later on with a class maybe, or a book you read, or maybe somebody that showed you something interesting? Thank you.
Thank you. Well, I was always interested in science. I always liked the science subject in school. Then specifically on the microbiome, that was because of a course I followed during my master's, in which a professor who became my master's thesis and doctoral thesis supervisor talked to us for the first time about the human microbiome. And to me, that was really like something very interesting. I mean, I was fascinated by seeing how all these microbes that we mostly see as something harmful for us are also living within us, and they're actually, I mean, we cannot live without them, and that they are so important for our health. But at the same time, we know so little about them. So from that point, I was okay. I want to study this.
Yeah, I understand that that curiosity there, because when I
Yeah,
heard about it, I thought, oh my God, I wish I was in that field.
cool.
So yeah, I totally understand. And for this specific project, you wrote a little bit on your website how you became interested to work in this, but do you have maybe some more behind the curtain story how this project came about, and how it was successful to get grants, and or how
Mhm.
people came together to do this project? Thank you.
Okay, so for my PhD, as you said, I studied the microbiota brain axis, which to me was just like something shocking. Like, how can the microbes in our gut interact with our brain? We There was evidence of gut to brain and brain to gut communication, but it was, I mean, hardly unknown how this could happen. So that's where I studied during my PhD. Uh, but then, toward the end of my PhD, I was seeing how the microbiome, especially the gut one, was linked to many diseases. So it was very important for our health, but we are missing this fundamental question on how do we get these microorganisms. Um, so that's when I decided to move to Italy for my postdoc to join Nicola Segata's lab, who was developing the tools to do strain tracking, which is the method that we need to assess microbiome transmission. And that's how I got there. I applied for European funding. I was successful in getting that. So then we started this project in like in a very ambitious way, trying to get as many as many samples as we could. Then we get more than 9,000. Analyzing all of them was not easy. It was many hours. But that's actually what we need if we want replicable results. So I'm very happy that so I joined that in 2019. So it was, I mean, more than 2 years of work for the analysis part. Then we have one more year for writing the paper, submission, different revisions, and so on. But in a bit less than 4 years, we finally got it published.
Well, congratulations, and I'm so glad everything worked out in that way, because now we have this wonderful research and more knowledge about our microbiome. So thank you for doing that, and the stage is yours, and everyone. The slides are pinned on top of the room. Feel free to access them, and um, the microphone is yours, Marie.
Okay, so thank you very much again for the invitation and for the kind introduction. I'm going to talk mostly about this research that we last did on the person-to-person transmission landscape of both the gut and the oral microbiomes. So first, a bit of an introduction. I'm on slide 2 now, on the human microbiome. So that's a collection of microorganisms that reside in and on the human body. So all the surfaces of our body that are in contact with the exterior have their own microbiome. And this microbiome is a collection of microorganisms. These are mostly bacteria, but there are also archaea, viruses, protists, fungi. But bacteria are the majority, so that's what we study mostly. And there are so many that we have in our bodies, as many bacterial cells as human ones. This ratio keeps being revisited, but we are close to 1 to 1. And in the gastrointestinal tract, in the gut, that's where we have the highest densities. We have on average 10 to the 12 bacterial cells. These numbers change based on from person to person. Also, in several diseases, this microbial load is reduced. And mostly we care about the microbiome because it plays a key role for our health. So disturbances in this microbiome have been linked to most gastrointestinal diseases like Crohn's, so colitis, celiac disease, but not only. also Alzheimer's, diabetes, depression, all have been linked to alterations in the composition of the gut microbiome. So now we know that they are very important for our health, so much that we could not live without them. And yeah, going to slide 3. So for the last decades, we've been studying this role of the microbiome in maintaining host health. And we now have some good knowledge on what factors modulate its composition. We know, for example, that what we eat is very important, as our microbiome changes during our life. So this is very important because in contrast to our genetics, we know we can modify it and try to keep it in a health-promoting state as much as possible. So yeah, care besides our genetics, it has a limited effect also about our lifestyle, about our diet, about the mode we were delivered. So babies born by vaginal or by C-section have a slightly different microbiome composition. Also, medication affects it. Our age, we keep accumulating microorganisms during our life. But then this diversity decreases when we get older. So there's this decline. And then also diseases, of course, influence its composition in a strong way. However, and in contrast to many pathogens, like for SARS-CoV-2, that we got to know in relatively little time how it spreads COVID-19, we know very little on how we acquire such microorganisms that are so strongly associated with our health and with diseases. So we also really know very, very little on how they are transmitted among individuals and spread in populations. Going to slide 4 now. And we think that this is not only a missing fundamental question, but also one with intriguing consequences. As a main big one, and that's what a lot of media channels put attention on, is that through the microbiome, non-communicable diseases, so those that are not linked to an infectious component, could become communicable. So if many of these diseases are strongly associated with microbiome composition and the microbiome is transmissible, they could in theory be transmissible through the microbiome. And of course, if we understand how this transmission works, we can also try to find ways of altering it. So in this way, we could try to deliver better microbiota modulations like pre or probiotics or fecal microbiota transplantations based on their composition. So looking at the strains of bacteria that are best at colonizing a new host, but also their timing. So what is best to actually do these interventions. Slide 5 now. So what do we know so far? There is a bit known on vertical transmission of the microbiome. So there are 3 main modes of transmission that we can think of. One is vertical transmission from mother to baby. The other is horizontal among peers. So vertical transmission was the first to be studied. And now we know that the first colonizers of our gut, the first bacteria that get there, are those from our mothers, followed then by microbial succession processes. And we studied this with our lab in some papers that were published as a proof of concept some years ago, in which we're looking at samples at different time points from mothers and their babies. On slide 6 now. So there it's when my colleagues started developing the methods to do this strain resolve metagenomics, because you need very high resolution to be able to see if actually 2 people have the same bacteria, because this is the way that we can use to infer if there was a transmission event. So if there is the exact same bacterium in a person and the other, this does really happen extremely rarely in individuals that have not been in contact, and it only happens for the same individual. So by building these phylogenetic trees, and when seeing that these leaves of the tree, so this is where we have the arrows, are very close to each other, almost flat, then we can infer a strain transmission event. So we started developing the methodology here. Now going to slide 7. And we saw an intriguing finding. That was that strains that would come from the mother were more persistent in the baby gut as compared to those from other sources, environmental strains. Suggesting that maternal strains would be much better adapted to the baby gut as compared to others. However, in infants, we don't see many of the typical species of bacteria that we find in adults. So we must get them later on. And that's what we started studying then. Another project in which we were involved, that's in slide 8, was seeing whether this transmission could also be detected across multiple familial generations. I was in Flanders, in Belgium at that time, and we set this multi-generational family cohort with families with from 3 to 5 generations of women. So we had 24 families with a bit more than 100 women, spanning a very wide age range, so from 0 to 98 years of age. And we saw there that, well, by applying these strain level profiling methods, I'm going to slide 10 now, that transmission would not occur frequently across multiple generations, but sometimes it happened. So this was actually possible. Going to slide 11 now. But then this question came to our mind. So are we actually seeing vertical transmission or horizontal? So is this the result of a strain going from the great-grandma to the grandma and at delivery, then when the grandmother delivers the mother, passing it and so on? Or are these strains just being transmitted later on, as we're seeing? Because especially we saw a very strong effect of kinship, so being in the same family for sharing the same strains, but even a stronger effect of cohabitation. So we had some clues that sharing an environment, living in the same household, would play a role. And that's why we started studying horizontal transmission, so that among peers that shared, that would not be happening at delivery, but at any other point in life. So slide 12 now. What do we know about horizontal transmission? So for a decade now, we know that individuals that cohabitate, also non-kin ones, so that they don't share genetics, share some microbiota similarities. Of course, this could be because they are eating, similar foods, following similar diets. But 3 years ago, in 2019, there was a paper published in which they demonstrated horizontal transmission in the microbiota in a small set of households in the Fiji Islands. And then there were also some hypotheses on how this transmission of the microbiome, of the gut microbiome mostly, could take place, and what kind of bacteria could be transmitted across different ranges. So, slide 13 now. We designed, as I was saying when I joined Nicola Segata's lab, this large-scale study of microbiome transmission to try to know better, in more detail, how this transmission takes place. We profiled more than 800 species of bacteria between the gut and the oral environment. So we study gut metagenomes from fecal samples, oral metagenomes from saliva samples. So overall, we collected a lot of fecal and saliva samples. And we also tried to use any data that was publicly available, as these were resources that had already been generated. So we tried to exploit them the best way as possible. And as I'm showing the map here, we also tried to have a global representation. So as in many other fields, microbiome research has mostly been focused in Europe and in North America. But we try to have a bit of a more global representation. Of course, it's not perfect yet, and we have still many countries missing. But to get a better representation of human lifestyles and geography. And in there we would include any samples that had pairs of individuals that were in contact, so mothers and offspring, individuals living in the same household, adult twins. That was also something very interesting for us. So these are pairs of twins that, of course, live together until more or less they become adults, and most of them would not live together anymore. So we then going to slide 14. Did also put a strong focus on the methodological work. So we tried to be very sound. So we had to see when we could say there was the same strain in two samples. So because of course all our work was computational. So we are getting these samples, we are we're accepting the DNA from there. We are sequencing all this DNA, and then we have to make sense of them. An important question here was, okay, when do you consider two bacteria that are found in two samples are similar enough to consider that there was a transmission event? I'm not going to get too much into detail here, but there was a lot of focus on having these methods as accurate as possible. And that will be so. Going to slide 15 now. So an interesting gradient. So we saw samples from the same individual, the gut microbiome panel now, which share the median 87% of their strains. So if I give a sample now and another one in 3 months, or we're doing here less than 6 months in general, a lot of the strains would still stay. So, and we know actually the microbiome tends to be very stable, except if there was some perturbation, like an antibiotics course, for example. And this was followed by mothers and offspring pairs, then individuals in the same household that share 12% median of their strains, then adult twins that would not live together anymore, or individuals in the same village, so that they share some environment but not as close as cohabitating. So these were, in both cases, 8% as a median. And finally, individuals in the same population or in different populations that would never cross each other, then there is a zero strain sharing rate. So 0 of their strains shared, which makes sense. On the oral microbiome, we saw even more strain sharing, together with some distinctive patterns that I'm going to discuss later. Slide 16 now, so step by step. I'm first going to talk about the gut microbiome on vertical transmission here. So what did we see was that mothers with their infants, up to the first year of life, they shared 50%, so half of the strains, which is a lot. Then, together with the expansion of the infant's motor activities, and also the accumulation of species from other sources, then the strain sharing between the mother and the offspring started decreasing. But this was long-lasting, as we had access to this last box in the box plot here in slide 16, which were individuals up to 85 years of age, together with their mothers. So these individuals from 50 to 85 years, they still share more with their mothers than with mothers of other individuals, meaning that this vertical transmission of the gut microbiome is very strong and also very long-lasting. Okay, we also saw that there was an influence of delivery mode, as could be expected. If we're getting the first colonizers of our gut from the mother during birth, of course we expect something different will happen when this birth is by C-section. But actually, these differences would not be apparent anymore after 3 years. So infants born by C-section would share less of their strains with their mothers early on, but then this difference gets compensated. Going to slide 18 now. So next we looked at horizontal transmission. and especially on households. And we see there that when we looked at older infants, so we did not see any significant differences between mother offspring, father offspring, between siblings, or between partners. So everyone in the same household tended to share, display similar strain sharing rates, but they all share much more than with individuals in the different households. So to us, this very strongly supports an influence on environment for microbiome transmission. Then we looked at twins, and we saw that strain sharing between them declined sharply with the first years they lived apart, much more strongly than with their age, which showed that not sharing an environment anymore was much more important than the fading influence of the mother at birth. So again, strong point for horizontal transmission. And then genetics played some effect, but a mild one. Sorry. These zygotic twins tend to share less strains than monozygotic ones. Going to slide 19 now. So our next question was, okay, how long? So how can this be this range of transmission? Do we need to be very close with someone to share strains with them, or also with someone in our village? We can just, can we, by their microbiomes, can we see someone that with whom we might cross and not someone we don't know at all? And we saw that there was, mild, but some strain sharing also. So some gut microbiome transmission within populations, which allowed us to build these networks, and this is something that is very often used for pathogens to track their spread. But here we can see that actually we can also track the spread of bacteria in our microbiomes, which is something very interesting for us. Then we went on and looked at transmission of the oral microbiome, so from saliva samples. There, as I was saying, we saw even more strain sharing than for the gut. And this is something that can be expected. The bacteria in our mouth have saliva as a very convenient transmission vehicle for them. But also here we saw some interesting patterns. So in there, we would not see this effect of inoculation from the mother at birth, but strain sharing was time dependent. This is that people that were older would also share more strains. So the more time you spend together with someone, the more strains you share. In general, mothers shared a bit more than the fathers with the babies, but in both cases, this strain sharing accumulated with time. Going to slide 21 now. So our next question was, okay, we are looking at transmission of the microbiome. We saw it's very highly transmitted. But actually, are all the species transmitted equal? Or are there some bacteria that are dispersal specialists? And we saw that indeed, so some species were better than others at being transmitted. But their transmissibility, their ability to be transmitted, was highly consistent across data sets, which to us points us transmissibility being a specific trait of this bacteria. So we tried to assess it better and to see what bacteria would be more often transmitted across each transmission modes. And we saw, for example, that for highly vertical transmitted species, so species that were very often frequently transmitted between mothers and babies, were bifidobacteria, which are very typical bacteria in the infant gut, but also less usual bacteria like Bacteroidetes or Kermansia, bacteria that has been strongly associated with obesity. And then in households and also in populations, so the bacteria were mostly transmitted horizontally, we saw many uncharacterized bacteria. So many of those, they did not even have a full name. So you see here some code, which means that these are bacteria that we are studying with metagenomics. So with computational methods, we can identify a genome of a bacterium, but these bacteria have never been grown in the lab, so we know very little about them. And seeing that these are among the ones scoring as most highly transmitted tells us that we should really study them and see what they do to our health. And something similar happened for the oral bacteria that were most transmitted. In there, as I was saying, for the oral microbiome, we saw mostly horizontal transmission. And actually, we also saw there that some species were consistently transmitted and highly transmitted in the different modes. So there are these three bacteria here. Also, they have a code. They don't really have like a clear species name, that are very poorly characterized. And these are the three ones that are most commonly spread. So we are now trying to study them better in the lab. I'm going to slide 23 now. So our last step was trying to see if some phenotypical properties of these bacteria, of the ones that we know, or for the ones that have never been cultivated, we tried to predict these properties based on their genomes. So seeing if those properties would explain their transmission dynamics. And we saw some patterns. So it seemed that gram-negative species, so these are this is the classification of bacteria. Gram-negative ones tend to be more resistant and tend to survive better in the environment. They need to be better transmitted between both mothers and babies, so vertically and in household. But then, if we looked at the species that could be transmitted among people in different households, so these longer ranges of transmission, these were mostly spore-forming bacteria. So bacteria can form spores as a resistant mechanism for survival, and also mostly were aerotolerant. So many of the species in our gut are anaerobic, which means that they just die in the presence of oxygen. So this is something that makes it difficult for them to be transmitted. But the ones that would tolerate oxygen, or tolerant ones, would be much more often transmitted between individuals in different households. And this is another study in which we also assessed what were the determinants of microbiome engraftment in fecal microbiome transplantation, because we saw there that when FMT is performed, the more bacteria from the donor engrafted in the recipient, the better the clinical outcome would be. That was also a large study in many different countries with different diseases. And we saw also there that mostly gram-negative bacteria, and in general, properties of the bacteria that would determine their survival would be associated with their better engraftment. So we are seeing some consistent results here. So, and it's actually a nice control for us, because when we look at person-to-person transmission, that's the joint effect of for bacteria of surviving the environment and being able to colonize a new environment, a new host, so a new gut. In contrast, for FMT, we are putting artificially these bacteria there directly, so they just need to colonize. Of course, they are there in a sick person, so there might be some different properties. But these are two different scenarios for microbial transmission that we think are very interesting. So overall, my take-home messages for you, going to slide 25 now, is that we said the microbiome, first of all, is very extensively transmitted among individuals in close contacts. It is transmitted vertically from mothers to infants, but it's not only our mother what will matter, because there is also then very large horizontal transmission. So sharing an environment plays a key role, and the individuals that are close to us are great reservoirs for these bacteria, and are the ones that are giving the bacteria that will colonize our bodies. We should, of course, there are different microbiomes in our bodies with different properties. And indeed, we saw distinct patterns for the gut and for the oral microbiome. And finally, microbiome transmission is not left to chance. So there were some species that were mostly dispersal specialists. So, and that was linked to some phenotypical properties that were transmitted more frequently. So we know still, as I was saying, very little about them. And we are going to try to study this in better detail, as this could actually help us design better, so more efficient probiotic treatments. With this, I would like to thank you all for your attention. Going to the last slide, slide 26 now. And I would also like to thank all my colleagues and also the collaborators that helped in this large-scale study. Thank you.
Thank you so much for sharing this really amazing work with us. This is really so interesting, and as I mentioned briefly before, it's like such a systematic, large-scale study that is really interesting, and it's really wonderful work. So thank you so much for sharing, and it was for sure a lot of work also. So
Yeah, thank you.
I see Susan is here, so I wanted to give her first the opportunity to speak. So hi, Susan, go ahead, please.
Hi. So I'm not in the sciences. I'm a filmmaker, but I found this incredibly fascinating. And I had some questions, but mainly some thoughts. So I just thought maybe I'd ask you, you might have covered it or you might have not. What happens in close proximity during living in close proximity, but involuntarily? Like, for instance, in prison, or if you are in a detention center or juvenile detention center, or if you're in school, or a situation where it's like traumatic. Had you done any research in that?
Okay, that's a very interesting question, and it's also great to discuss our research with people from different backgrounds, and that was a very nice opportunity of yeah, after publishing our paper. So, I mean, we don't. I mean, of course, everything I'm going to say now is speculation because, yeah, we in our study we study people that live together in the same household, mostly voluntarily. We expect. We didn't really look at the prison setting, but I would expect. I mean, we don't really have a saying on how our microbes want to spread. So I would expect them to, I mean, take the most advantage they can of this new proximity, and then just spread also there with the other individuals that they would have in close contact.
Very interesting. Could I just follow up with one more question?
Yeah, sure.
In terms of viruses, were there any thoughts in terms of, like, because of the pandemic and the things we're going through, and possibly additional viruses and viruses that are not responding potentially to antibiotics, in the issue of superbugs, is this something that is going to make this even more pivotal and even more important in terms of the immune system?
Yeah. So I have to say, yeah, our research focused mostly on bacteria because these are the most, most of our microbiome is made out of them. In that case, there is, of course, some of these bacteria in our microbiome that are associated with our health, others that are more disease associated. And to us, that was something already interesting to see that it seemed there was a slight association between more pathogenic bacteria having more aggressive colonization strategies. We then look at bacteria that were resistant to antibiotics, but that's an interesting follow-up. We are now looking more at the role of this transmission in different diseases. So also viruses is something definitely to follow up on.
Thank you so much. This is incredible. Thank you.
Thank you. Thank you very much for your interest.
Yeah, thank you so much, LT, and yeah, Lennon. Welcome to the stage. LT, please go ahead, and then Lennon.
Okay. Can you hear me all right?
Yep.
Yes.
Okay, great. Thank you. A lot of work, I have to say. Thank you. So I wanted to ask, I'll ask a first question, then I reserve second question after Lena, maybe.
Okay.
So, yes. So on slides, where was that? Slides 21, you said that are all gut species transmitted equal? It's not, right? Your answer is that. So
Yeah.
I'm asking, you have so many different data sets. You have North America, you have Europe. What about the geographically?
Yeah. This
Thank you.
It's a very good question, and something I did not cover too much in detail because of time. To us, it was something interesting to see that there was a smaller effect of geography or lifestyle than what we would have expected. So it seems that in general, like bacteria are, being more or less transmissible is a characteristic of the bacteria themselves, and they just don't care where we are. But it's actually something that people are asking because we know that individuals in non-westernized societies, which mostly based this classification, of course, it's not very clear cut, but it's a way we can look at lifestyle. So in general, individuals that would not take antibiotics have less access to drugs, eat less processed foods and mostly more fibers. We know that they have a more diverse microbiome, and that the more diverse the microbiome, the better we are. So that's something known on the composition as a whole. But if we look at the transmission of the bacteria, it seems that the transmission from the mother and from other individuals in the household is similar. So this extra diversity in non-westernized societies, it probably comes from other individuals or from the environment.
Okay. Then London, if you are ready to ask, you can ask. Otherwise, I will follow up with your twin. You mentioned twin
You can
studies.
follow up. I have kind of a two-parter too. So go ahead, please.
Okay, good. Then because you also mentioned, I forgot which slides you mentioned, like twins. You have a data set with twins. That they got the same data set. Not them, same data. They got the same kind of from the mother, and then they may not necessarily live in the same household. They go over, one in the, I don't know, China, one in US, or one in Europe. Let's say triplets. So nowadays you say many. What kind of things, at least, what can you tell us about that? Yeah, you mentioned it, but I didn't hear much more.
So the twins, the pairs of twins that we got were mostly from the UK. So there is a very large study that has been going on for a very long time now. It's the Twins UK study, in which they follow huge numbers of twins. And there, to us, that was an interesting example because for twins, we can assume they got more or less the same microbiome from their mothers at birth. So seeing that, and we saw that when they got older, they would share less of their microbiomes, which makes sense. I mean, this influence from the mother is fading, and they are exposed to other bacteria from other sources. But much more stronger than their age was when they started to live apart. So this to us was clear proof of the transmission among them. So twins would get more or less the bacteria from the mothers at birth, but then they also keep exchanging bacteria when they live together and less when they live apart. Then I also talked about the influence of genetics, as it's been shown that monozygotic, so identical twins, share more of their microbiomes as compared to the zygotic twins, not only because of the moment of birth, but also, probably because of an effect of genetics. And there we saw, as I was saying, a mild effect. So in the age categories, when there was a significant difference, that was monozygotic twins winning. So having higher strain sharing, but that was not so strong. So again, this shows that environment, so direct transmission is more important for our microbiome than genetics. Did this answer your question?
Yeah. I think I was going heading that direction. So are you saying that the microbiome in our guts actually they probably have some influence on, because you mentioned like what is this, twins, identical twins versus non-identical twins?
Yeah, exactly.
Yeah, thank you. Okay, I'm done with my question for now. Thank you very much.
Welcome.
Yeah, I guess the thing that struck me most curious was the intergenerational, the vertical transfer of microbiomes. And you had said, and I think I can see in the slides, although it's very small, an orange bell curve that is, basically one generation, you've got a whole lot of fidelity, that mother to offspring transmission. But after that, like it goes down precipitously, like by second generation out or third generation out is quite a bit less likely that you're going to have that transmission of microbiome fidelity for that many generations. And so I'm just wondering, is that all due to household and proximity, or in changes? I guess what I'm asking is, like, if you were to have done this study in Belgium, like 1600, when people are much more local to village and where maybe their diets are not changing so much, like there's less selection pressures, would you expect that curve to be similar, or?
Yeah, that's a very good point. So this was, for us, so we had this question because in mice, it's been, of course, their generation times are much shorter than us, so it's much easier to do this kind of studies. And we're trying to see if there was this transmission from great mama to grandma to the daughter and the granddaughter. Of course, we would look at all these samples at the same time point. So if we see there was transmission, we don't know if it was at birth or later on. And that's why we started having this idea of checking at the influence of the environment. And indeed, we saw that, with more generations, less strain sharing, but also when starting to live apart, their microbiomes changed and the number of strains that were shared decreased. Yeah, indeed, Belgium is a very specific setting. So, families tend to live in a very restricted geographical area. So children would not move very far away. They would also keep seeing each other quite frequently. So there, and indeed, the diet is also quite stable. So we could expect that in other settings, if one would move to a different part of the world, following a different lifestyle with different diet, then we would see less strain shared.
Great. And I think that actually more or less answers my second question, which was just to kind of take the geographic example to the extreme and say, well, what if you had a family, say in East Asia or Eastern Europe, who had a child, raised it to 2, then had to adopt it, say, to the United States, and you were to compare microbiomes of the native nascent family and the adopted family and that individual, like.
Yeah, indeed, there have been some published studies on adoption cases, because it's a very interesting setting to look at this. But that was mostly on the general microbiome composition. So it would be very cool to also track strains in those cases. Yeah, thank you.
Yeah, thank you.
Yeah, thank you so much for all these questions. It's really interesting. And then Elizabeth in the chat shared, if you take a probiotic strain that has very high engraftment rate, would you only have to take it once, or does it? I guess probably, if you take those supplements, is it similar? Sticky, like that it stays in your system, like human to human transfer?
So that's a very good question, and that would be the ideal case. The real answer is that, we just hope this treatment would be more efficient, because I mean we know that for now probiotics don't work very well or as good as we would hope for. And so yeah, if they have a much higher engraftment rate, we hope they will more stably colonize this microbiome and have a stronger influence. Of course, the microbiome is a dynamic environment, so I mean, taking it only once would make it hard for specific bacteria to stably colonize. But hopefully, we will need to take it less often or in smaller quantities.
Yeah, thank you. And do you, so my questions, as probably a lot of things were asked for the future, what's the next thing you're going to analyze? Because you alluded a little bit to antibiotic resistant treatment. Is there maybe a prediction study going on that you can maybe um, assess vulnerabilities in families maybe, and kind of in the future treat this, since birth or so? Or is that something maybe that would interest you study in the future?
Yeah, certainly, and and in general, association with disease. Our study was on generally healthy individuals as a first study to see how transmission of the microbiome takes place in normal situation. Now we want to look at association with disease, especially on diseases that run in families. See what's the role of the microbiome. So if microbiome transmission would have a role in also transmission of the disease, this would be something certainly important for us. And the another point is other sources of transmission. So far we looked at individuals in close contact, but we think that, for example, also pets could be interesting reservoirs. So just trying to expand the picture of transmission more, both in general and in disease settings.
Yeah, that's interesting. I think that will be really interesting in the future to learn about disease states. And do you think, like, this is very, very speculative, but we have kind of trouble, especially in mental health disorders, since your previous work was kind of involved in mental health. Also, we have kind of trouble making a genetic link. It's a very low percentage of the genetic component.
Yeah, indeed.
Do you think that the microbiome will hold a better causality relationship than genetics? It's yeah, I don't know.
Yeah, that's what I would hope for. And in some cases, it seems so. Like, as for the microbiome itself, I mean, genetics play small role, but not, I mean, just a small one. So hopefully, yes. I mean, of course, I think these diseases are multifactorial. The microbiome will not be the only answer. We'll have to, design treatments, trying to focus on the different factors at once. And microbiota modulations might help, for example, the current, increase the efficiency of the current anti-depression treatments, for example. I don't think they will replace any therapy, but they could hopefully complement them to get better efficacy.
Yeah, interesting. And then would you be interested to see how maybe different microbiome structure could also affect gene expression in the human? Because there's a lot of epigenetic gene expression work going on, also related to disease and health and aging and so on. So do you think that maybe the microbiome would influence that too? And are you also thinking of looking into aging, if people with a specific microbiomes maybe age healthier than others?
Yeah, thank you. These are two very interesting points. So on gene expression, definitely. I mean, I would expect that the microbiome affects host, so human gene expression in the gut, but maybe not only, and also in other tissues. And then for the aging, yes, I mean, there are some studies, and it's interesting to see that during senescence, so in elder, so toward the end of our lives, um, in line with this immune senescence, we are also losing diversity of our microbiomes. So maybe there could also be some room for microbiome treatments trying to promote healthy aging. Then, there are also some studies on assessing, cohorts with very longevous individuals, seeing if microbiome composition plays a role, and it seems, that it has something to do with that.
That's really interesting. I did know that that in aging there's such a difference in the microbiome. So I think this will be really interesting to follow your work in the future, because all of these are really important questions, and and if you can answer just a subset of them, that will be making a huge difference in people's lives. So yeah, I think, is there anything you would might want to add that maybe we didn't think of asking currently? That's probably my last question.
Okay. Well, thank you very much again. And yeah, I think we mostly covered the part of microbiome transmission and some of its implications. As in diseases, of course, there is a lot more on the microbiome in different situations, but that would be a whole different talk. So I think it's good to close it here for today. And again, thank you very much for the invitation and for the nice questions.
Yeah, thank you so much. I agree there will be, involvement in cancer and in every aspect of our lives. So, yeah, thank you so much for doing this work, and we really appreciated you coming here and sharing it. And I hope we'll hear you again one day and talk about your future research. So, thank you so much,
Great.
Mireia.
Thank you. Thank you very much.
And thank you everyone for coming and asking interesting questions. It's always way more interesting when people come with their questions that they have, because it makes the discussion more have more levels of interest. And our next talk, um, if you are interested in discussions like this, will be with Dr. Martin next week, with origins of bilaterian life cycles. So basically, it's evolutionary biology and how male and female life cycles developed, and it will be really interesting to learn about it, I think. And yeah, again, thank you, Mireia. Enjoy the rest of your day, morning, evening, wherever you are, and I'll close the room in 3, 2, 1. Bye everyone. Thank you.
Thank you.